Clock on my watch, so we will go ahead and get going. Good afternoon, everyone, or evening. My name is Victoria Ortiz. I'm the Community Engagement Manager with the Tahoe Regional Planning Agency, or TRPA. And tonight we are going to give an overview of the Tahoe Keys Control Methods Test. So things that we've learned in the last year, which was year 2 of the test, and what's coming up or what to expect in year 3 of the test this summer. So we do have 3 great presenters for you tonight. We have Dennis Sabaglo with TRPA, Dr. Lars Anderson with TKPOA, the Tahoe Keys Property Owners Association, and Laura Patton with the League to Save Lake Tahoe. Now, as you probably already noticed, you are automatically muted. If you do have any questions, please pop them into the Q&A, which you can see at the bottom of your screen, and then our panelists will go ahead and answer those, or we'll save them and answer them live at the end of the presentation. We anticipate the presentation taking about 40 to 45 minutes, and that leaves 15 to 20 minutes for questions. So again, if you do have any questions throughout the presentation, please put them into the Q&A, not the chat. With that, we will be recording this presentation, so it's already recording, and we will post this to the website, which is tahoekeysweeds.org, and then we will also make sure that everyone on our Tahoe Keys e-news gets the link and the slides. So with that, I will welcome Dennis Sabaglo with TRPA to start the presentation.
Great. Thank you, V, and thank you, everyone, this evening. I appreciate your continued interest in this project and helping us find solutions to control weeds in the Keys, but ultimately protecting Lake Tahoe. I'm Dennis Sabaglo. I'm the Aquatic Invasive Species Program Manager for TRPA. I'll just do a brief overview here momentarily, and then I'll turn it over to Dr. Lars Anderson. He's a science consultant with the Tahoe Keys Property Owners Association that will go more into the details of what occurred this past season and what we expect to see this coming treatment season. And then Lars will turn it over to Laura Patton from the League to Save Lake Tahoe, who will give a perspective from the league. And before we get started, I'm sure some of you have heard about the lawsuit that was filed, and with a ruling from the judge. Just wanted to address that briefly, and that Hunt is continuing to review that decision and considering their options. But it's the understanding of this group that the judge did not enjoin TKPOA from conducting the non-chemical methods testing of this test. And since aquatic herbicides were only considered and approved the first year of the control methods test, TKPOA is proceeding with the 3rd and final year of this important test that utilizes only those non-chemical methods such as UV light and bottom barriers. Once the test is complete, all that data that was collected over the 3 years of this test will be reviewed holistically to inform what set of tools will control the infestation for the long term. And for this next year, TRPA will continue to conduct that independent monitoring of the test. Next slide, please. So just a brief reminder of what the Tahoe Keys Control Methods Test is. It's an innovative approach. We're looking at multiple techniques that are used here in Tahoe, some pioneered here, such as UV light, and then others, non-chemical methods with bottom barriers and diversissed suction, but also the one-time use of aquatic herbicides. And that approach in and of itself is innovative. No one has ever tried to implement aquatic invasive species or aquatic invasive weed control project with a one-time use of aquatic herbicides, followed up by all non-chemical methods. And ultimately, we're trying to find solutions for the long term to treat this high priority infestation that's truly the main source of aquatic invasive weeds for the rest of the lake. Next slide, please. So why are we concerned about aquatic invasive species? The main 2 we're looking at in the Keys are Eurasian water milfoil and curly pondweed. They are 2 notorious aquatic plant invaders throughout the country, and they've been established in the Tahoe Keys for several decades. But why do we care about them? They have severe impacts to Tahoe's delicate ecosystem. They outcompete our native species, such as Elodea. It's a native plant species. They provide habitat for other invasives, such as large and smallmouth bass or bluegill or crappie, which are fish species that do not belong here in Lake Tahoe, and they prefer those habitats that weeds create, making it warmer. They can concentrate excessive nutrients when they die off. They certainly reduce our clarity that we're famous for in Lake Tahoe, and it fouls the nearshore, ruining the experience of all of our visitors. And with Lake Tahoe's $5 billion recreation-based economy, controlling aquatic invasive species is one of the highest priorities for the region. Next slide, please. So just a little background or reminder of where we've been on this project. 2019 started a significant public engagement process. We wanted to ensure transparency and having lots of public input ahead of the project as to what should be included in this test. And that was another outcome of that engagement, is that we should start with the test. A comprehensive environmental analysis was done that included lots of baseline data. And that led to approvals from the TRPA and Lahontan boards to start with the CMT in the year one in 2022, which included the use of UV light and aquatic herbicides in an initial knockdown period, what we call Group A methodologies. And then there was lots of resource protection measures to ensure any impacts from whatever treatment method was employed was not going to impact the lake. And then subsequent to 2022, we started the project in 2023 with just the Group B methods. That was the second year of the test, so that we just completed. Those are all those non-chemical methods we mentioned before, such as the UV light, bottom barriers, laminar flow aeration, and DASH. It's diver-assisted suction hand pulling, where divers are able to pull specific plants so they can differentiate potentially between a native plant and an invasive plant. And now we're moving in.
Oh,
yes.
sorry. Dennis, just do you mind speaking up a little bit? Some people said that it was a little hard to hear you.
Oh, sure.
Thank you.
And now moving into 2024, we're looking at the third and final year of the test that will essentially mimic the 2023 activities with the non-chemical methods, as I mentioned before, UV, bottom barriers, laminar flow aeration, and the diverse assisted suction hand pulling. And then once the test is complete, there's lots of data that has been collected over those 3 years that will be compiled, and looking at the overall efficacy, what treatment combinations resulted in a 75% reduction in biovolume, and what set of those could be implemented for the long term to control this infestation for the long term, for the duration. That would also include additional environmental analysis, whatever that projected full-scale treatment might be, and then having the scientific partners look at those data and the treatment proposals to make sure we're looking at something that's scientifically based. Next slide, please. Again, this is the map of the control methods test. All the polygons are the identified treatment areas, looking at the Group A of UV light and aquatic herbicides in independent and combination approaches to get that knockback. And then Group B methods, you'll see a map here momentarily, were implemented in year 2, which is all the non-chemical UV light, bottom barriers, laminar flow aeration, and the others I mentioned, to maintain a 75% knockback. And that was achieved in multiple locations, and that will be continued in year 3. And again, continuous monitoring and data collection throughout the lifespan of this test. Next slide, please. This is an example of a map for the Group B year 2 and year 3 treatments. You'll see the polygons of the original treatment locations are still there, with now some smaller polygons or shapes that identify the different Group B treatments within those, and those are representing the areas that had a good percentage, or that 75% breakdown or knockback of aquatic invasive plants, implementing those other methods to maintain that knockback. And the important part of this test is being able to adaptively manage or adaptively react to what conditions are seen in the lake, or in the Keys, I should say, in addressing and putting these treatment methodologies in those areas where they're needed. And I think with that, I can turn it over to Dr. Anderson.
Thanks, Dennis. And thanks very much for joining us on this webinar. I'm going to cover a few things this evening, a little bit about the first year, because this is an adaptive process, as Dennis mentioned. And so the results we get from the first year and second year, each one sort of drives what we do in the subsequent years. And I'll talk a little bit how the Group B methods are implemented, the effectiveness of those last year, a little bit on the nutrients and water quality impacts we have. And I will spend some time toward the end talking about the differences in the water levels from our first year in 2022 and the tremendous change that occurred last year and this year as well in the water levels in the Keys and how that impacts the plants that are growing there. Next couple clicks, please. Dennis mentioned a couple of the Group B methods. I wanted to show you a close up on the DASH method, which is the diver-assisted hand harvesting. And the fellow on the right is just coming up from hand pulling plants out. That black tube on his off his shoulder is a suction tube, and he physically removes the plants by hand from the sediment, puts them in that suction tube. It's all underwater, and they get drawn into that green bag that you're seeing in the lower left image there. And that bag is a mesh bag which collects all the fragments and all the plants that are brought up by the divers. And when that bag gets full, they haul it out of there, put the contents in that blue pail. You can see it's sort of behind the cage there. And then they start it all over again. So it's a time-consuming process, but it actually has some real advantages because it physically removes the invasive plants we're interested in removing. And if the water is clear enough, the divers can actually leave the native plants like the Elodea that Dennis mentioned behind. So that's kind of the approach. And I wanted to show one other shot here of a plant. Some of you may not have seen this, but if you're in the Keys right now, it's doing pretty well, unfortunately. This is curly pondweed. And I wanted to show these dark areas in the image. Those are called turions. They're actually not flowers and they're not seed. They're kind of a vegetative structure that the plant forms every late spring and summer. And each one of those turions will form a new plant. And one of the reasons this plant is so invasive is those spread around when the plants get moved around by water currents, by boats, or anything else, move them around. And that's why we're seeing a rapid spread of this plant. It's also in South Lake Tahoe along the South Shore as well. But the key to controlling this plant is to prevent it from making these turions and then to control those turions when they sprout in the fall and in the spring later on. So I'm going to show a little more details about this. Next slide, please. And Dennis kind of covered the background. I want to point out we are in group in our year 3 of the methodologies for the CMT. And again, only the first year included the use of aquatic herbicides. The subsequent 2 years, last year and this year, are non-herbicide approaches. I want to focus a little bit on the monitoring just for a minute. Dennis mentioned we're doing a lot of intensive monitoring, and we are. And one of the key questions on the CMT is, what do these methods do in the way of controlling our target aquatic plants? And of course, do they affect the other non-target plants that we want to maintain? And just to give you some example of the intensity of sampling for the aquatic plants, the macrophytes as we call them, each of those sites you've seen in the slide that Dennis showed you with the different CMT sites, each one of those is given 30 rake samples every 2 weeks. And by the time we're done for each year, there's over 8,000 rake samples taken from spring to fall in these sites. So we have lots of intense and very useful data. The rake samples are important because they tell us not only how much of the plants are there, but which ones are most prevalent, because when the rakes are pulled up, the crew actually identifies the plants and what percentage they make up on that rake. So it's very useful information. The other thing I'd mentioned at the bottom of the slide, we use these hydroacoustic scans, which are kind of like a very high-end fish finding device. Uses the sonar signal to determine what's on the bottom of the water, which includes the plants in the bottom. And we use that to determine a biovolume, or the rough estimate of the quantity of plants that are there. And I'll show you a couple examples of those. Once we have that scan data, that information can be translated into what we call a heat map, which shows you where most of the plants are. The more intense the color, the more plants that are there. Next slide. So just to show you the difference between year 1 and year 2, based on these scans that I just mentioned, On the left-hand side is August 30, 2022, which was the first year, midsummer of the first year we did our treatments. And the left-hand side, area A, was primarily herbicide treatments. And you'll notice that those are all blue and green, which means very low biomass in those areas. The middle section of that is either our untreated reference sites, which had no treatments, or it was the LFA, this laminar flow system that we're using. The area B was a different kind of herbicide used over there, which looked pretty good too. And the lower left-hand side of the left panel shows area C, which is Lake Tahoe. And that's kind of interesting. That was our treatment site, which looks really good because it's all pretty much blue and green. Then you move to August 25th, last year, 2023, and you can see that even though we didn't use herbicides in the second year, the effectiveness of those herbicides kept the plants down to very low levels in that area A and in area B. But you can also see that in the untreated areas in the middle and in our LFA sites, we had lots of plants. That's with the yellow and the red areas in midsummer. But you'll notice the Lake Tahoe, and I put an arrow to show that we kind of lost control of that one area. And we lost control because mostly what happened in Tahoe is this one of our plants called coontail seemed to move into the sites, and it's the only plant that we're working with that has no roots at all. So it can be moved around by wind currents, by boat action, and primarily by wind that actually moves it around. So probably what happened there is we had control, but it moved into that site in the subsequent year. But this is the kind of information we can get. And these, by the way, these scans are done every 2 weeks as well. So you can see that the intensity of monitoring for the effectiveness of these treatments is very high. Next slide. So from year one, what we did learn from year one is that the indithol herbicide controlled all the target species, especially Eurasian water milfoil, which we almost eliminated in those sites. It was more than 95% control, very encouraging. The other herbicide we used in year one was triclopyr, which is only really expected to control Eurasian water milfoil. And it did a very good job of doing that. It doesn't control coontail and has some effect, but not much on curly-leaf pondweed. The reason we use triclopyr is because it moves it, that herbicide actually moves into the roots and rhizomes of the Eurasian water milfoil, so it can provide a longer-term control. In the third bullet down, you see we had good control with our UV light in most target plants, 50 to 75% reduction. This is a unique approach, operational approach for UV light. It was less effective on coontail, partly because coontail forms a very dense mass, and sometimes that mass actually shades out some of the UV light so that the top might get controlled, but below that may not get the effective exposure that it needs. There's also some difficulty in getting treatments in between docks in the site. Dennis mentioned Elodea. What we found in the first year that within our herbicide treatments, we actually had some increases in Elodea, our native plant in those sites. That's primarily because we were able to reduce the competition from our invasive plants in those areas. But the good news was in year one, we had sufficient control of our target plants to warrant year two and the use of these non-herbicide group B methods. In other words, we had 75% reduction in biovolume in most areas. So the most successful treatments in year one were the two herbicide treatments, the UVC light, but we had very little effectiveness from our laminar flow aeration, that bubbling system that we used there. Next slide. So we had a decision to make at the end of year one, and I just circled in the orange here, at least on my screen it's orange, that we had both target plants and our native desirable plants coming up in year two. And that led us to use the group B methods that are listed below: the bottom barriers, the UV light spot treatments, and the diver-assisted hand removal, as Dennis mentioned. So using that information that we got from our rake samples last spring and fall, we determined which sites should have the group B method treatments. Next slide. So Dennis showed you the slides from the first year. This is our map showing the overall sites. Those are the large polygons and all the sampling points on there. The next slide, please, shows you the one that Dennis showed you. And I want to point out the fact that the group B methods are relatively small compared to our large sites that are shown in the larger polygons. Also in this site, the crosshatch sites are all our untreated reference sites. And you see we had some in Lake Tahoe as well as the West Lagoon in these situations. The other thing we tried to do with the group B methods was to repeat our treatments in several of the sites, so we had some replication of the bottom barriers, the dash treatments, and the UV spot treatments. So if you look at this now, you'll see within those sites that we had treated in year one, most of the sites had some group B methods approach to control and to sustain that 75% control. Next slide. The actions in last year were pretty critical to the program, of course. We did conduct our spring macrophyte samples a year ago. And from those samples, we could assign which group E methods should go where in the sites based on the amount of plants that were coming up and what species were coming up. We installed the bottom barriers using 3 types of materials to see which ones would be more easily to work with and which ones were more effective. We had UV spot treatments. We also started our UV treatments in our what we call our combination sites. These are sites that in year 1 only had herbicides along the shoreline treatments, not in the middle of the areas of those sites. So in year 2, we started our combination UV treatments in the middle sections of those sites that were only treated in the shoreline with herbicides in year 1. And then of course, we started our diver-assisted hen harvesting, and we actually expanded the use of the laminar flow aeration to 3 sites, including some sites in Lake Tahoe. And of course, we continued the monitoring. The only difference between year 1 and year 2 monitoring is that we didn't have to do any more herbicide monitoring in year 2 because we'd already shown that we were below detectable levels at the end of year 1. Next slide. So in a very simple way to look at the effectiveness of year 2 with the group B methods, I put together kind of a chart that shows a pretty simple arrangement of whether we actually met the 75% criteria or we didn't, or we partially did. So any arrows that are upward and are green means we've met the 75% sustained reduction in the sites that were treated with bottom barriers that are listed here. And you'll see we have several sites. Again, these are replication sites. And you'll see we had some problems with one of our triclopyr sites, site number 14 toward the bottom, that orange arrow that goes to the right. Some of these difficulties were again, because coontail moved into these sites and was not easily controlled because it kept shifting around in those areas. But for the most part, the bottom barriers were pretty effective. And I should point out too, that at the end of the summer, the bottom barriers were removed and we continued to monitor those sites. And one of the things we learned last year that is important in terms of managing curly pondweed is that even though we had control while the bottom barriers were in place, once they were taken off, some of those turions that were under there started sprouting within a few weeks after the barriers had been removed. So now we're looking at a possibility of a later treatment in late summer for that kind of a situation. Next slide. For the diver-assisted removal, you see we had pretty much success across the board. Again, some of these areas that are shown in the orange arrows that are horizontal, that was primarily because the coontail moved into these sites after the divers had removed them. The other problem we have with physical removal is because you still have plants growing next to these sites, and they can actually grow into a site that has been removed by the divers over several periods of time, several weeks after they've removed the plants initially. So, but for the most part, it worked pretty well. We learned some lessons about this, and that is to make sure we have large enough sites with the dash approach to keep the plants cleared out for the season. And this year, we're going to go back and probably do more repeats than we did in year 2. Again, we had some success in LFA sites where we actually had the divers removing plants. And remember, the LFA really didn't work very well by itself. So we had some good control with the plants once we added the divers removal to those sites. Next slide. So this last one shows the effects of the UV treatments. First of all is the spot treatments. These are smaller areas within the large sites that I mentioned before on that map. And again, most of these worked really well. The only problem we had was one of the LFA sites that didn't sustain its control very well. But actually, this looks pretty successful across the board, whether it's the UV spot treatments, UV combination treatments, which we had started last year. Those were the middle zones of those sites. And then the UV only sites, which is area site 22 and 24. I should mention that site 23 was not treated last year. It is back on the treatment schedule for this year. And that's why that arrow is going to the right. It was not really treated, so it wasn't included as part of the treatment response in the second year. Next slide. So based on those results and all the rake samples and all the scans that we did from year 2, we can kind of summarize the results this way, that the 2022 first-year treatments of endothal and triclopyr in those sites retained excellent control, especially for Eurasian water milfoil. And it was pretty much absent in all the sites that we used the herbicides in, except for the controls, that is the untreated reference sites and the LFA sites. And some of that newly submerged shoreline that I mentioned, because the water levels were much higher last year than in our first year. We also had the increase of our desirable native plant, Elodea, by about 20% last year. That's encouraging. And the repeated dash treatments sustained 75% control of our target plants in most of the areas. The bottom barriers kind of had a mixed result, and that's partly because once the barriers were removed, some of those plants regrew in there. Some of the treons resprouted. So that sort of tells us that we need to do something about the post-removal of the barriers to continue controlling those sites. Water quality was a little bit better in year 2 than year 1. Water clarity was better in general. And part of this has to do with the large amount of volume of water that we had in because of the high snowpack in year 1. But again, the LFA approach did not really affect our target plants. It may have some effect on algae, but right now what we can say is it's, at least to this point, it's not doing much to control our target aquatic plants. Next slide. I wanted to show you some of the reason that we use rate data so much. It's very useful. And that's a lot of data here, but I'll just guide you through the bars. First of all the blue bars are the data from our untreated reference sites. So they've had no treatments whatsoever. They're sampled on the same schedule every 2 weeks with rake samples at all the sites. In this case, we're looking at the triclopyr site, the one that was treated with that one herbicide. And on the left-hand side, you can see this is the abundance of plants based on how much plant materials are on these rakes. And if you look at just year one on the left-hand side, you can see the mid-channel, which is that upper bar there, we had pretty good control of our plants with the triclopyr herbicide. It got more than half, 50% control on a lot of these, and some was down to 60 and 75% control easily. But if you look on the right-hand side, you'll see in year 2, we had some control, but not as well. It didn't carry over as well in the second year from that first year's treatment. But the most important difference on this graph, I think, is showing the difference between the mid-channel and the shorelines. And if you look at the bottom set of graphs there, that's all from the shoreline data. You can see in the first year we had pretty good control, but in the second year we really did not. There's not much difference between the blue bars and the green bars. In fact, a lot of the green bars are much higher than our controls. And this probably had to do with the fact that we had plants growing in shoreline that was not even available in year 1, and it was available in year 2 because of the water level change. So there's some big differences in the mid-channel and the shorelines. And that's another lesson from, I think, our last 2 years of work in the Keys. Next slide. So this is the LFA site that I mentioned, the laminar flow aeration site, just to bring home the message that we didn't really see much difference between our controls, the blue bars and the purple bars, which are the samples from the LFA sites. Again, this is the abundance of plants on those. Basically, the bottom line is this treatment really has at least we've seen no effect on the aquatic plants we're trying to control. Next slide. That was 26 I just showed you. This is a sample of curly pondweed taken from 26 last month. Just to emphasize that the LFA really is not affecting, not really reducing the growth of curly pondweed. That's curly pondweed. That plant is about 1/2 meter or maybe 2.5 feet tall. And they're really growing quite well in that site. So we have good evidence that that particular system, that method doesn't really have much of an effect on reducing our plant growth in those sites. Next slide. So again, looking at abundance, I wanted to bring home this message again with the endothol treatments from the year 1 only to start with on the left-hand side. Again, blue bars are reference untreated sites. All the red bars were the sites where endothol was used in the first year. And you can see we had tremendous reduction of plant biomass or abundance of plants in those. Even in year 2 on the right-hand side, you can see that we had a great sustained that reduction of the plants with the endothol treatment that was only done in year 1. So this year 2 is with no additional herbicides at all, nothing. But again, look at the difference between the mid-channel and the upper bars in year 2 and the shoreline. This also underscores the problem we had in year 2 with higher water and more territory, if you want to call it that, that the plants can grow in. So a lot of this difference we're seeing between mid-channel and shoreline is because those shoreline plants never even got treated in year 1. None of those were even exposed to cover with water, so they couldn't grow. So that probably explains some of the differences between the mid-channel and the shoreline areas. Next slide. So I mentioned the importance of rakes and being able to tell these differences. This is our UV site treatments. Again, the blue bars are untreated reference sites. This is UV sites. This is site 22, which was continually, repeatedly treated with UV. And you can see we had some pretty good effect in mid-channel if you look at the year 1 and year 2. But again, in the shoreline, because it's very difficult for this equipment to get in between docks, especially when boats are moored there, It just can't do as good a job in the in-between area dock areas as it does in the mid channels. So that's another lesson we've learned from year 1 and year 2, and we're going to do some adjustments on year 3 because of that. But you can see the difference again between the mid channel and shoreline effectiveness of UV light. Next slide. So the rakes allow us to tell what species are prevalent and what are not. So on the left-hand side, you see I've asked the question, how often does Eurasian water milfoil occur in a sample? And again, the data you're looking at is from hundreds of samples, year 1 and year 2, actually thousands of samples. And this is from the end-of-thaw treatment sites. And in this case, again, the blue bars are untreated reference sites. The red bar are the very few instances where your Eurasian water milfoil was actually found on a rake sample in both years. So I think what this tells you is that not only did we reduce the amount of plants there, but just their frequency of occurrence was reduced by those treatments. Not only in year 1, but also the effectiveness of that year 1 was carried over into year 2 as well. Even in the shoreline, we were still pretty effective with Eurasian water milfoil reduction in year 2. So that's encouraging. You know, Eurasian water milfoil is still in Lake Tahoe. It's being controlled in various areas. But it was the #1 problem in Lake Tahoe for up until about the early 2000s when curly palmy was first found. But we're doing a good job with this one. It looks like there's a way to control this and do a really good job at reducing its impact in the Keys and then probably in Lake Tahoe. Next slide. So for year 2 monitoring, just a couple of things to mention. We'd look at all kinds of parameters with water quality, the pH, dissolved oxygen, and so on. And the thing to remember is when you have a lot of plants growing, they tend to elevate the pH above levels that you'd like to have out there. So when you have overabundance of these submerged aquatic plants, the pH goes way up. What we saw in year 1 and a little bit in year 2 is the areas that we had treated and successfully reduced plants, we had lower pH, not too low, but lower and bringing it down below where it should be. Typically, though, in our controls, we had pH that was over 8.5. By midsummer due to large growth of plants and algae in those systems. The other thing we've noticed, even in our untreated reference sites, was low oxygen near the bottom. This is pretty common when the Keys or any water, body of water doesn't move much, it doesn't get much circulation. You tend to get less oxygen in the bottom, and we saw that. But again, I mentioned before that the water clarity in year 2 was better. That our turbidity was lower. We measured this frequently as well. And it generally increases in midsummer, but overall last year was much clearer water than the year before. And right now it's looking like year 3, this current year, 2024, is even clearer than last year, probably again due to high water levels coming in. And the good thing about year 2 was we also saw very few incidents of the harmful algal blooms, HABs as we call them, and they were less severe in 2020 than they were in 2022. So they're encouraging results from that. Next slide. So to compare the conditions, just really want to bring this home because the water levels do make a difference. They do affect how plants grow. They don't affect all the plants in the same way. But in year 1, we had very, very low levels of water. We had shallow conditions. We had warmer conditions in the Keys. We had our curtains up that prevented water from moving around very much because of the need to keep herbicides within their confined areas. And so we wind up with high turbidity and persistent detectable levels of triclopyr, not a good thing. Year 2, totally different story. Click on one more click there, thanks. Year 2, 4 feet more water in the Keys than our first year in 2022. And as I mentioned before, not only is that deeper water, but it also submerged about 10 to 15% more shoreline than the first year. So that 10 to 15% more shoreline was shoreline that was never treated by any method in year 1. And that's where we're also seeing this high level of plant biomass or abundance in those near shore areas that were not even exposed to any treatments in the first year. The other thing that deep water does, it cuts the light down. So typically for a lot of these plants, they need some light. They don't need much, but they need some. And what we found out is below 12 or 13 feet in the Keys, there's barely enough light for these plants to grow rapidly. And that's why I think we had fairly low growth, even in our control sites in year 2, for that deep water. So we had lower biomass of those control plants as well. So next slide. So now we're into year 3, and I'll finish up here really quick. We even have more water. We have 2 feet more water at this time this year than we had last year. So compared to the first year, we have 6 feet more water in the Keys. And maybe a few more percent of shoreline is submerged this year than last year, but most of that change occurred between year 1 and year 2. But with the water clarity we have in late May and June is excellent. This promotes plant growth where it can, and we are seeing a lot of curly-leaf pondweed growth, which it seems to do pretty well in deep water with good clarity. And we're seeing a lot of these turions now being formed on curly-leaf pondweed, those ones I showed you in the first slide that allow this plant to spread so rapidly. So the take-home message, in addition to the treatments and the responses, is that this typical change in water level has some tremendous effects on plant growth in the Keys and in Lake Tahoe in general. And it's one of those things that we're going to have to integrate into long-term planning and to have different scenarios, perhaps, for different conditions of snowmelt and snowpack and water depths in the summertime periods, because they really do affect what plants are going to grow there and how dense they get. Next slide. So just to summarize what we did in this current year coming up in year 3 with our grouping methods, we increased our bottom barriers by about 18% to encompass more areas that we see plants coming into. We increased the diver removal areas by about 24% from last year, but we really increased a lot of the UV spot treatment areas by 130% because we're seeing some effectiveness with this treatment in these smaller areas where we've got new plants coming up. Based on our harvest that we're done, our monitoring that was done in the last month and a half or so. So we are responding in a very adaptive fashion to this. And the next slide kind of summarizes this as well. Next slide. And you can just click through those. Thank you. So for the these UV treatments, we've got additional boat coming in this year. We added our Site 23 I mentioned to the treatments, which will be done every 3 or 4 times during the season. We've increased the spot treatments in the mid-channel areas, and we're probably going to be using some spot treatments on the bottom barriers when they're taken out in the fall. This is a new approach that we're hoping will work because we did see that these turions can sprout once those barriers have been removed. Next click, please. Thank you. The bottom barriers, as I mentioned, almost 20% increase. And these were actually installed a little bit earlier in May and June. And we're going to keep these on a little bit longer than we did last year in these sites, just to keep more of control of the plants. And for the diver system, we started earlier this year. We're adding 3 more sites. Again, some repeats, probably 3 times in some of those sites as well. And we're also getting more data on how much of the turions from this curly pondweed is coming out of the diver removal operations. And lastly, again, just like with the UV system, we may apply the dash method in these bottom barrier sites once the barriers have been removed, to compare and see how effective that is in removing these sprouting curly pondweeds. So we're really using a lot of adaptive methods. Every year we learn something new and we apply that to the next year. And at the end of all this, the next slide, as Dennis pointed out, is to develop a integrated long-term integrated method that actually sustains control in a very effective way. And I think with that, I will turn it over to Laura from the League to Save Lake Tahoe. Laura?
Thanks, Lars. My name is Laura Patton. I'm the Natural Resource Director at the League to Save Lake Tahoe. I'm just going to give you the League perspective on the control methods test and discuss a little bit about our role in really maintaining the progress and focusing on containment in the Tahoe Keys. Next slide, please. So for the last 67 years, our mission at the League has really remained the same. It's to protect Lake Tahoe's cobalt blue lake for future generations. Next slide. When most of us think about Lake Tahoe, we think about a clear, pristine lake and that beautiful blue color. Next slide. However, aquatic invasive plants, as we've heard from Dennis and Lars, really threaten Tahoe's blueness and pose the greatest ecological threat to the lake, which could turn Tahoe's waters a murky green, as you see here. Next slide. Because of this threat to Lake Tahoe, the League has really prioritized aquatic invasive species control lake-wide, not just in the Tahoe Keys, as aquatic invasive plants really threaten not only the Keys, but the entire lake. As you can see in this 2022 map of the established and treated infestations throughout the lake, you can see that they pose a threat not only to the Tahoe Keys, but marinas and shallow areas all around the lake. Next slide. So in 2023, the league recognized and really officially prioritized aquatic invasive species control in the Tahoe Keys as a keystone campaign, which it continues to be today. And you can go ahead and scroll through those animations. Sorry about that. As part of that campaign, we've really deployed citizen scientists since 2013 to start collecting water quality data, first as part of our Pipe Keepers program, and that program later evolved into a program called Eyes on the Lake. And so we've really focused on aquatic invasive species control, prevention, surveillance, and monitoring throughout the last decade. Next slide. Over the last decade, we've really committed to the deployment of new and innovative technologies to address the weeds in the Tahoe Keys. So we've been involved in the Tahoe Keys, every step of the way, from funding new technologies like bubble curtains to contain aquatic invasive plants, to monitoring that water quality, to also measuring the efficacy of different non-herbicidal tools such as laminar flow aeration, while also advocating for the best available science to be deployed to really address the threat of aquatic invasive species, not only in the Keys, but in the lake as a whole. Next slide. So more recently, the league has been involved in the control methods test to ensure that the robust scientific research and review is utilized throughout the test. So while it's in progress, we're also testing again those non-herbicide containment methods on the east channel of the Tahoe Keys and funding those efficacy studies on the TKPOA side of the Tahoe Keys while this test is ongoing. So this slide shows the laminar flow aeration, which is one tool that the league is monitoring to see how effectively this tool in particular breaks down nutrients in the Tahoe Keys. So this type of efficacy monitoring we find to be really important. It's in its second year to see if this tool specifically can break down the muck and those nutrients that are actually feeding those aquatic weeds. Next slide. So as we seek to contain the aquatic weeds within the Tahoe Keys while this control methods test is in its third year, the league is really focused on maintaining the progress that's made through not only the control methods test, but the deployment of other technologies to really focus on containing the weeds within the Tahoe Keys so that they don't go out of the Tahoe Keys, as you can see in this image from 2020. So we've really focused on the containment of aquatic weeds. It's important to protecting the lake from these aquatic invasive species. Next slide. So the containment that we're talking about is focused on both the east and west channels of the Tahoe Keys, as seen in this image, and the west side of, in that yellow circle that you can see, is really where the control methods test is focused. And we're focused not only on that aspect, but also on that east channel that you can see. Next slide, please. So the three technologies that the league is focused on to really maintain progress throughout the control methods test and containing aquatic weeds within the Tahoe Keys are the technologies we've deployed that are pictured in this slide. So we've funded and deployed, and we are conducting efficacy studies on what we call double bubble curtains, which have been deployed in the east and west channels of the Tahoe Keys. And those are deployed to really create a barrier from the lagoons to the lake to prevent the spread of aquatic invasive plants into the lake proper. So on the east channel of the Tahoe Keys, in partnership with EcoClean Solutions and the Tahoe Keys Marina, last year and this year, we've also deployed an aquatic Roomba that we call the Pixie drone, and a fragment and trash debris robot that's called the Collector. And those two aquatic weed-eating robots that you see pictured here work in combination with those bubble curtains in order to contain fragments. And containing fragments also helps stop the spread of invasive plants and prevents those turions that Lars discussed from spreading into the lake proper. Next slide. So this image really shows the proposed deployment of additional containment technologies in the East Channel. So while the control methods test is going on the West Channel of the Tahoe Keys, the league is deploying additional technologies on the East Channel as well to prevent that spread that you saw in that earlier image of the Tahoe Keys offshore. So really focusing on those containment technologies again to make sure that we're maintaining progress and containing the aquatic weeds. Next slide. So in the third year of the control methods test and beyond, the league is really focused on maintaining the progress made during the control methods test. We're also supporting independent scientific review by the Tahoe Science Advisory Council of each year of the test and also of the test as a whole as we move forward. We're also continuing to find and pilot innovative technologies to keep Tahoe blue. Next slide. Sorry about this. So in addition to those containment technologies, the league is also working to really educate visitors and locals alike to make sure that, you know, what has happened in the Keys in terms of aquatic weed spread doesn't become a lake-wide problem. So we funded technology to clean, drain, and dry non-motorized equipment. We have 2 machines currently, and those 2 pieces of equipment, clean, drain, and dry non-motorized equipment in order to not spread weeds to other areas of the lake. And then we've been conducting trainings all around the lake with that machine. And also we've been conducting trainings to help citizen scientists identify and report aquatic weeds through our Eyes on the Lake program. So, you know, as the league watches the control methods test closely to monitor progress, we're also focused on that containment, and we're focused on education, monitoring, and surveillance of the entire lake as a whole to keep Tahoe blue. And with that, I think I'm going to turn it back over to V. Thank you.
Great, thanks, Laura. So really just to summarize here, there is an incredible urgency to try to find solutions to the aquatic weeds in the Keys. And it has been a long journey to get to this point, and it's going to continue forward. So we do have thousands and thousands of data points and lots of data being collected every year and learning from this test in order to be able to inform a long-term solution. So as we mentioned here, finding a solution is critical. Next slide. And with that, we've come to the end of the presentation portion. So thank you all for sticking with us and for listening in on this lovely evening. At this point, if you have any questions, I encourage you to please pop them in the Q&A. We've seen a couple come through during the presentation and those have been answered, but I would love to open it up to our panelists if anyone does have questions. And let's see here. One just came in on what is being done to reduce the amount of fertilizers going into the Keys. So I don't know who wants to take that one, but maybe as part of it too, you can explain a little bit about where the nutrients for the weeds comes from.
Yeah, I'll take that. Yeah, so the Keys have restricted the use of phosphorus for several years now for landscaping in the Keys. And that's a critical important because mostly the limitation in the Keys is phosphorus levels, both for algae growth, but also for macrophyte growth, the weed growth as well. But nutrient inputs are multiple. They're coming in from runoff, street runoff from the Keys. And there was a question about nitrogen. Yeah, probably some nitrogen comes in there too, but I think there's been a lot of recent studies and flow measurements indicating that a lot of these nutrients and a lot of the inputs are coming in. First of all, there's atmospheric nitrogen is a huge input to the lake itself and also the Keys, but also the stormwater runoff is another problematic area because it's basically A lot of the streets around surrounding the Keys area they go right into the Keys, and that's a city drainage system, basically. So, but I think the Keys have really done a tremendous job in not only education, but also in terms of their regulations for homeowners of cutting down the use of fertilizers, and particularly phosphorus containing fertilizers in the Keys. That's rather important. But there are other inputs as well. And there's natural inputs from degradation. And the other point to not forget is that all these plants that are growing in the Keys, when they senesce in the fall, they're dumping all those nutrients right back into the system. So they are kind of a cycle. And if you can reduce that biomass of the plants in there, you have a chance of reducing it. The other thing we do know is that there's quite a bit of nutrients locked up, not locked up, but available in the loose organic sediments in the Keys. And that's another issue that has to be dealt with, I think. And there's ongoing discussions about how that could be reduced possibly.
Thank you, Lars. There's another question in here that would be good for Dennis, and that's what's being done about the New Zealand mud snail.
Yeah, thank you, V. Again, Dennis Abaglo, Aquatic Invasive Species Program Manager for TRPA. The largest part of the infestation that was discovered last year is more offshore of the Keys, but there were some detections after the initial discovery in the northern parts of the Keys, so near the channels that lead from the lake into the Keys. Unfortunately, at this current time, there's not viable treatment options, as we've discussed in many platforms, such as our governing board, but we've taken quite a bit of effort to delineate the infestation along the South Shore and to ensure that the implementation of the CMT is not going to spread any potential mud snails that may be within the Keys, to ensure various protocols to decontaminate in between sites, to ensure that there's no spread if there is a New Zealand mud snail in that work area. As of now, we have not discovered any New Zealand mud snails in any of the test locations, but we're still going to take those precautions to ensure we're not spreading anything if they do end up in those locations.
Great. Thank you. And if you do have more questions about New Zealand mud snails, what they are, what you can do to help prevent their spread, I would recommend going to TahoeBoatInspections.com. And there's a whole page on what boaters can do, what kayakers, divers, fisher folks. So that's a good place for learning more information. Thank you, Dennis. Okay, there's a question in here, probably again for Lars. Is dissolved oxygen better in the LFA sites?
Yeah, good question. And in fact, the difference between the dissolved oxygen profiles, that is where it is in the top and bottom of the water column, is most dramatic in the LFA sites compared to the sites that don't have any treatments of that at all, controls or otherwise. What we're seeing with the LFA is you get a more uniform distribution of dissolved oxygen. And keep in mind what's happening when those are running, you're getting a constant aeration from the bottom up with all these fine bubbles containing oxygen, of course, and air. So you actually would expect to have that more uniform, higher levels of DO in that system. And Madonna, that's exactly right. The DO, that's one of the major differences we're seeing, is the DO does create a more uniform distribution of, the LFA does, of DO in those systems than in the other sites.
Great. Thanks, Dr. Anderson. And another question, so don't go too far away here, but can the bottom barriers remain in place throughout their useful life?
Well, that's an interesting question. We are using barriers we used before. Depending on what the material is made from, they can last several years. It just depends on how they're stored and how they're cleaned and what happens to them underwater when they're actually in place. But some of these get torn just because you have to manipulate them and anchor them properly. So there's some variable lifespan on them, but most of them can be used at least 2 or 3 times. It just depends on what condition they're in when they're removed and how they're stored. That's really what it is. And what we're looking at now is which barriers are the most, I guess, the most easily deployed and most effective. That's kind of one of our questions to figure out.
Thank you.
I can add to that as
go ahead.
well. On the bottom barrier question, typically we see pretty significant dieback under bottom barriers within a few months. And so the strategy of taking them out at the end of the season gives native species a chance to rebound faster than the invasives, and then we can go back and retreat those areas if the invasives come back. There's also a challenge within the Keys that sediment could build up on top of them from the adjacent areas, so that could create just a implementation challenge that we're trying to avoid as well.
Thank you both for that. There's a couple questions in here about the lawsuit. And Dennis, I'm just wondering if you would mind touching on that again for folks who might have missed it in the beginning.
Yeah, certainly. As far as we understand, the lawsuit does not enjoin TKPOA from moving forward with non-herbicidal methods. And as you hopefully one of the things you took away from this is that there's a lot of data and information being collected, and we'll continue to work with our partners on that data to inform a scientifically based approach to treating the infestation for the long term, whatever that methodology may or may not be. And we'll continue to work with Lahontan and the Tahoe Science Advisory Council on what those next steps might be.
Thank you. And then there's a couple questions in here about what's going on with the circulation system and if it will be fixed. So that's probably a good question for TKPOA. And I know, Christine, you put an answer in the chat too, but maybe would you mind revisiting that verbally?
Sure. Yes, the Tahoe Keys is doing sort of a separate EIP evaluating the circulation system. It will need to be redesigned. It cannot be reinstated in the way that it is. And so we're evaluating what the construction modification should be, and then also the role that the circulation system will actually play within the Keys. Because the way it was used in the past is probably not the way we're going to use it in the future to combat sort of the issues that we have going on.
Thanks, Christine. And Lars, I'm seeing another question in here about if did LFA play a role in reducing the algal bloom that occurred in area A of the herbicide application?
Actually, no, because it's not really in area A. Area A, the improvements in water quality and in general in the West Lagoon, I think a lot had to do with the fact that we had 4 feet more water last year. And that was clean water coming in, and we had more circulation going on with it as well. No, I think what we're seeing with the LFA is, in general, the dissolved oxygen is more dispersed throughout the water column. I think the pH is a little bit more stable, but not all that stable. Keep in mind that the pH really does change every day, every night radically because of plant photosynthesis and respiration. So as long as you have plants growing in there in the summer, you're going to have elevated pH. But I think what the LFA does, it certainly does move water around, and it certainly does distribute the dissolved oxygen more uniformly.
Great, thank you. Okay. I'm seeing most of the questions are being answered here, and we are a minute out here from the hour. So I would say if you have any other questions, you have contact information here on the screen for Dennis Zabaglo, Dr. Lars Anderson, and Laura Patton. You can also find a lot more information at TahoeKeysWeeds.org. That's where we have all of the special reports that have been released, and you can find a lot of information on the project, where we are right now, what's happened, where we're moving towards. I want to say thank you again to everyone who joined us tonight. Hope you have a lovely rest of your evening. And again, it has been recorded, so we'll be sure to send out the recording and the slides to everyone following this presentation. So with that, I'll open it up to the panelists if there's anything else that you all want to say before we sign off.
Just thanks for your attention and your interest in the project.
Agreed.
We're working at it really hard, and we hope you can get some solutions soon.
Agreed.
Yeah, thank you.
Thanks, everyone.
All right. Well, with that, we'll give you back your evening. Thanks for joining us and have a good rest of your night.